So, the nervous tissue, the basic nerve cell that is usually called as neuron. The neuron is a specialized nerve cell that is the basic subject and basic point in related to the nervous system. So that you already know the cell is a structural and functional unit. So here we have to go for the today just the identification of this neuron. So here the neuron is main the two portion, one is the cell body and another one is processes. The cell body is contains the nucleus and cytoplasma, and the processes are the projections from the cell body, they are dendrites and axon. So usually the dendrites are short process and axon is long process. So the dendrite short process from the membrane, cell membrane, that is cell body, and axon is also arises from that cell body and it is long process. So just you see this figure, the cell body is the structure of the cell that is covered by cell membrane, and inside there is cytoplasma and as well as nucleus and the Golgi apparatus. So other organelles. Then again the dendrites, see there is small process which arises from the cell membrane. Then axon is a long that is also arises from this cell body, and that will be ends by a number of small branches that is called as telodendrite. So be remember of this the basic points. So what is the cell body? The cell body is variable size and the shape. The size and shape is variable, and that contains the nucleus and cytoplasma. So the nucleus is single nucleus and it is central and spherical and contains the large nucleolus. Then cytoplasma, it is a fluid that will be located within the cell body. So this cytoplasma is covered by a cell membrane, that is outer covering, that also called as the portion of cell body or the cell membrane. So within the cell membrane, that is inside, there is a cytoplasma. This cytoplasma is a fluid that particularly contains organelles like mitochondria, Golgi apparatus, lysosome, chromophilic substances, and also the nasal granules or nasal bodies and neurofibrils. So usually the nasal granules present in the nerve cell body and dendrites, but these nasal granules are not present in the axon. That is the point you have to be concentrate. The nasal granules or nasal bodies, they are particularly present in the nerve cell body and dendrites, but not in the axon. So usually these nasal bodies are involved in protein synthesis. The protein substances are very important in related to the normal function and the metabolic activity. So that is why the protein synthesis that will be especially managed or maintained by the nasal bodies. Then neurofibrils, these are a thread-like variable length. The thread-like structures, the variable length, they are also present in the cytoplasma. So these are the general explanation. Then again, what is the cell processes? So the cell processes is the dendrites. So these dendrites are short processes and these contain cell bodies and mitochondria, that the dendrites. The neurofibrils, they lack a myelin sheath and neurolemma. So neurofibrils, they have not covered by an myelin sheath, lack of myelin sheath, and also neurolemma. Then axons. The axon, usually neuron has a single axon. So there is a single axon which is long, slender, and it is a thread like a processes that particularly arises from the cell body at the region of axon hillock. It contains the mitochondria and neurofibrils but no nasal bodies. So this particularly axon is a thread-like elongated structure that arises from the cell body at the region of axon hillock, and it contains particularly the mitochondria and neurofibrils but no nasal bodies. This is the main explanation. Then what about the myelin sheath? The myelin sheath is the outer covering of the axon. The most of the axons are surrounded and covered by an insulating sheath, that is myelin sheath. It is a fatty myelin derived from the supporting cells, especially oligodendroglia within the central nervous system and Schwann cells outside the central nervous system. So the myelin sheath is a outer covering which will be surrounded the insulating sheath of the fatty myelin sheath. So usually it will be supporting by any different cells like oligodendroglial cells helps for the myelin sheath formation in the central nervous system. But in the outside the central nervous system, the Schwann cells helps to develop the myelin sheath. That is the point. The oligodendroglial cells in the central nervous system, Schwann cells in the outside the central nervous system. These cells are supporting cells. They are called as supporting cells. Helps to form the formations of myelin sheath, and that myelin sheath is just covers the axons. It is an insulating layer. Helps to the fast and the transportation of the impulses very clearly. The myelin sheath is not continuous, but it is interrupted in middle or in some regions. So that myelin sheath is particularly no continuation, but it is a break, it is a cut down. So that particular cut down portion by a constriction, that is called as node of Ranvier. So the interruption of the myelin sheath continuous, that interruption, that constriction is called as node of Ranvier. So just to be observed here, so this is the just structure of the cell, so the different neurons, but here we understood the cell body, the small processes are, that is dendrites, and this is the axon. See here the constriction, that means gap of the myelin sheath which will be covers this axon. So that myelin sheath covering, that's an interrupted, that means break. So that portion is called as node of Ranvier, that is cutting up the myelin sheath. So the myelin sheath is covers the axon, particularly in the peripheral system. Suppose if it is in the central system, this myelin sheath is formed by the oligodendroglial cells. So this is the just picture. So what you have make it, but this is different neurons, that I will tell you the classifications. So what is that? So the neurons are classified as a unipolar neuron and pseudo-unipolar neuron, then bipolar neuron and multipolar neuron. So the main classifications of the neurons, usually there will be three types. The unipolar, pseudo-unipolar is almost the same. Some text and some explanations, somebody are explaining as an unipolar and some they are explaining as a pseudo unipolar as a separate, but almost these two are similar feature. So in the unipolar, what will happen, you can see this figure. So the cell body has a single process. That single process further divided into two divisions. Among that, one becomes a dendrite and one becomes axon, that is unipolar neuron. So this the unipolar neuron, you can see the cell body, it has a single process, then it is divided into two divisions. Among that, one becomes dendrite and one becomes axon. So that is the point you have to be explanation. Then bipolar. In the bipolar, what will happen? The cell body has two process, and that one process becomes dendrite and one process becomes axon. This is called as bipolar neuron. Then third one is the multipolar. The multipolar neuron, as the cell body has a numerous more number of processes, short, they are called as dendrites, and one long process that is called as axon. This is multipolar neurons. So majority the multipolar neurons which will be present in the most part of the motor and internuclear connector neurons. But this unipolar neuron particularly, that is in the fifth cranial nerve and also dorsal root ganglion of spinal nerve, that is the spinal nerve dorsal root. That dorsal nerve root in the spinal cord and the spinal nerve, that is having a pseudo unipolar neuron. Then bipolar neuron usually in the eighth cranial nerve, and multipolar neuron, that is motor and internuclear portion, that is connector neurons, that means center neurons. There you will get the classification of these three types, and you can observe this figure. First one is unipolar, means single process. Bipolar, the cell body has two process. Multipolar, more than two process, that is usually multipolar neuron. That is the main classifications of the neurons, and in some different regions they have located. Then another type of, that is Golgi 1 and Golgi 2. What is that Golgi 1 and Golgi 2? So the Golgi 1 is long axon. If the axon is too long, then it is called as Golgi 1. If it is a short one, then it is called as Golgi 2. This is simple explanations, but depend upon the length of the axon, that is Golgi 1 and Golgi 2 type of neurons. That is the main classifications. So be understood. So this is unipolar, and this is bipolar, and this is multipolar neuron. So these are the main classifications: uni, bi and multi. Uni means single process, bi two process, then multi more than two process, that will be the divisions of neurons is there. Then again neuroglia. So what do we mean by neuroglia? The neuroglia means the supporting cells. They are not neurons main, but they are supporting to the neuron. So these supporting cells are usually called as neuroglia. So we remember the supporting cells to the neuron and to the system, that group of cells, they are called as neuroglia. So these neuroglia that consist of the different type of cells. First one is astrocytes. See this astrocytes, this cell is star-like appearance. It is just a star-like appearance, that is called as astrocytes. Astro means that is star. So this star-like the cell, that is astrocytes. This has divided into two classification. One is protoplasmic astrocytes and fibrous astrocytes. So what happens here? In the protoplasmic astrocytes, there is a thick process that is present particularly in the grey matter. The process is that is more thickness, and this particularly located in the grey matter of the central system in the brain and spinal cord. Then these fibrous astrocytes, they are thin, they are longer, that present in the white matter of the brain and as well as spinal cord. So internally the protoplasmic astrocytes present in the grey matter and fibrous astrocytes present in the white matter. So these are thin and longer process, but in the protoplasmic thick and they are mostly, that is the process, the structure. Then another one is oligodendrocytes. So here oligodendrocytes, that is a smaller and few processes and present in the both matter of central nervous system. So both matter means grey and white matter of the central nervous system, oligodendroglial cells. So there is a small and few number of process. Oligo means less, so smaller and few processes, that will be the oligodendrocytes. Then next one is the microglial cells. These microglial cells, they are a preventive mechanism, that means phagocytotic cells. They prevent the microorganisms, defense mechanism. So these phagocytic cells, they have a scanty cytoplasma and have a few processes and lies in particularly in the grey matter. So these are usually phagocytic cells, microglial cells. So these cells help to the preventive mechanism. So these are the main three oligodendroglial cells. Apart from that, some of other cells, they are also called as supporting cells. For example, ependymal cells. So these ependymal cells are columnar cells. They are lining the cavity of the central nervous system. The cavity of the central nervous system means the ventricles. All the ventricles, they lined by the ependymal cells internally. These are columnar cells. That will be the ependymal cells. Helps in the cavity of the ventricles. First or a lateral ventricle, third ventricle, fourth ventricle and central canal, that portion. Then Schwann cells. These cells are helps for the development of the myelin sheath, especially in the peripheral nervous system. That axon is lined or covered by a membranous portion that is formed by the Schwann cells, that is myelin sheath. So these are called as supporting cells. Ependymal cells and Schwann cells are the supporting, and others also supporting cells. So be remembered here, usually the neurons have only limited power of regeneration. So usually what will happen, neurons, they have a limited power of regeneration, that is particularly in the early life. That means this would be around the time of birth. The cell bodies of most developing neurons lose their mitotic apparatus like centriole and mitotic spindles. On account of that, after the birth, the cell division process is not continue, it will stop. So whatever the cell division process is there within the embryo life, within the mother in the portion of that child level. Once the birth takes, after that birth, the mitotic division is disappears. So then further there is no cell division of the neurons. That is the point, very important, be remembered. Whatever the number of the cells at the time of the birth, the same number of the cells after the birth till the end of life. That is the point here. Then again, the axons in the central nervous system are myelinated by oligodendroglial cells. So these cells don't form neurolemma to assist in regeneration. That is another point. That means the axons of the central nervous systems are myelinated by oligodendroglial cells. These cells don't form the neurolemma to assist in regeneration. But the axons in the peripheral nervous system that have a neurolemma that can be repaired as long as the cell body is intact. The axons in the peripheral system that have developed as the neurolemma layer, so that layer is helps for the repairing of the long, that is still how long the axon is connected to the body. That is the important point. Then similarly, the growth of the new axons will not occur. So there is no new axons development. Only if the gap is at the site of the injury is too large, or the gap becomes filled with the dense collagenous fibers. So only there will be a possibility of the repair of that axon if the gap is very less. If it is too long, then it will be also difficult. So that will be filled with the collagenous fibers. That is the point one important. Then proteins assist in the regeneration of the damaged axon. The protein molecules that will be supporting to the regeneration of damaged axon. So the damaged region is rapidly converted into special form of scar tissue, that is astroglial proliferation. That scar tissue forms a barrier to the regeneration. That is the point. But you be remember, however, the function is never completely restored after a nerve is involving. That means if the nerve injury, the axon injury, however, it will be a limited repair and it have a the functional concept. That means it cannot store as earlier it has. There is some changes in the functional concept. Then once damaged, there is a permanent damage. We remember, once the neuron, especially the cell body damage, usually almost it is a permanent damage. No chances of regeneration, no chances of new cell formation, almost it will be a stop. So this is a very important in related to the neurons subject, because in other cells there is a regeneration, the cell division process is there, but in this neuron no cell division, no processes usually after the birth portion. So this concept we have to make it clear of the region. And one more point you have to be remember, the short processes are, as you know, dendrites. So these dendrites transmits the impulses towards the cell body. These dendrites, these are transmits the information, impulses, that sensory impulses towards the cell body. Then axon is transmits the impulses from the cell body to the peripheral portion. So that is the functional concept of the dendrite and axon. We remember the dendrites are a short process as anatomy. These dendrites transmits the impulses towards the cell body. The axon is long one and that transmits the impulses from the cell body to the peripheral region. That is the functional concept. So this is about the neurons and cell structure. That will be the basic points of the nervous system. I hope that you are clear about this.